Membrane receptors, G proteins, cyclic AMP, and signal transduction. A receptor is a protein molecule to which another protein molecule binds. This attachment can cause an array of different effects to the cell. Activities in the cell can be determined by the receptors that are present or not present within the cell membrane. Metabotropic receptors are receptors that lack a channel that opens and closes and uses neurotransmitters as a ligand. These type of receptors link onto G proteins. A G protein is a cell membrane protein that functions as an intermediate between hormone receptors and effector enzymes. They are also called a heterotrimeric G protein because they consist of 3 subunits, an alpha, beta, and gamma subunit. Many different types of G proteins exist, but each become activated through binding to a receptor in the cell membrane. This graphic will show the process of the activation of the enzyme adenylyl cyclase. The purpose of adenylyl cyclase is to convert ATP to CAMP. As you can see, this conversion takes place in the inner surface of the plasma membrane. Cyclic AMP is a second messenger, which means that membrane channels may be opened or closed in order for this activation to occur. Step 1. In this step, epinephrine will act as our ligand, the neurotransmitter. Epinephrine binds to the receptor, which triggers an effect. This binding also initiates protein conformation. Step 2. Guanosine diphosphate is converted to GTP, guanosine triphosphate. It then causes the alpha subunit to dissociate from the receptor and attach to the effector. This triggers the formation of cyclic AMP from ATP. After the alpha subunit binds to the effector, adenylyl cyclase, this triggers the formation of cyclic AMP from ATP. Cyclic AMP binds to the regulatory subunits of protein kinase A. The catalytic subunits then dissociate from the regulatory subunits. This process is called phosphorylation. The effects of phosphorylation on the cell depend on the characteristics of the protein that is phosphorylated. Cyclic AMP activates kinases, which is an enzyme that joins a high-energy phosphate group to another molecule. This process is called phosphorylation. The effects of phosphorylation on the cell depend on the characteristics of the protein that is phosphorylated. Now we will discuss the process of deactivation. The GTP is hydrolyzed and converted back to GDP. This deactivation causes the alpha subunit to dissociate from the effector and rejoin the beta and gamma subunits. Adenylyl is now deactivated. Therefore, the production of cyclic AMP from ATP is halted. The adenylyl cyclase is now inactive. Phosphodiesterase then comes in and removes all remaining CAMP. This causes the catalytic subunits to reattach to the regulatory subunits in protein kinase A. This means phosphorylation has stopped.